Hi, my name is Annie, and you're watching Keeping Up with the Crystals using X-ray crystallography to solve for the protein structure of mSIRal. The first question that might come to mind is, what exactly is a protein structure? And secondly, why would we want one? Well, each protein in your body is a string of amino acids folded uniquely to perform specific tasks on a molecular level. By figuring out the unique folds of a protein, we learn about how it interacts with other proteins and molecules to perform those tasks. Such information is of clinical significance, as it 1. Gives insight onto how the protein interacts in key molecular pathways, and 2. Allows us to design therapeutic drugs in a process called structure-based drug design. The protein that we want to crystallize is the mouse equivalent of a human immune protein called mSIRal, hence mSIRal. We're specifically interested in mSIRal as it might serve as model organisms in the initial stages of medical research and drug trials. A recent study demonstrated that both human and mouse mSIRal were important for our immune response against infections, with mSIRal being identified as playing a role in controlling herpes and salmonella infections. This is highlighted in the presented graphs, where the gray bars represent normal cells and the red ones represent cells without mSIRal. As you can see, for both herpes and salmonella, the red bars are higher than the gray bars, indicating that without mSIRal, there is a greater degree of infection. Thus, discovery of the mSIRal protein structure poses to be an important first step in mSIRal-related medical research. We can determine the structure of mSIRal using a technique called X-ray crystallography. In X-ray crystallography, you grow protein crystals in drops of buffer and shoot them with X-ray beams. This generates a diffraction pattern on a piece of film. From this diffraction pattern, you can glean an electron density map, which then you can use to solve for protein structure. Preparing for X-ray crystallography necessitates 4 major steps. First, you need to generate enough protein stock. Using this stock, you screen for conditions that may promote crystallization of the protein. This includes subjecting your protein to varying pHs, temperatures, salt concentrations, etc. Once you find a condition that seems promising, you try and optimize the conditions to grow larger crystals. I've already generated enough mSIRal protein stock, as indicated by the highlighted band on the gel. Using my mSIRal protein stock, I've also set up upwards of 900 unique conditions and monitored them over the course of 2 weeks to identify hits, aka conditions where it looks like a crystal may be forming. Lucky for me, I found a hit on my first day of observation, with a small crystal I've dubbed Chris. To generate larger crystals, I've replicated my hit conditions in a larger drop format. Once again, on the very first day I observed crystallization, with the appearance of 2 new members of the crystal clan, Kendall and Kylie. We will continue to monitor their growth as well as fine-tune the concentration of precipitants in the future experiments to see if the crystals can grow any larger. If we successfully grow large enough crystals, we'll harvest them and ship them to a synchrotron, where they'll be diffracted with an X-ray beam. In the long term, we hope that we'll be able to solve for the crystal structure of mSIRal and use it to inform future studies of medical significance. Of course, none of this would have been possible without the support of the Morehouse Lab, especially my graduate mentor Irene and my fellow undergraduate Emma. That's all I have time for, but until next time, thank you for keeping up with the crystals.